Welcome back. Today we're going to be talking about the cell cycle and mitosis. So, cells actually dividing. So, first, some fun facts. All humans are roughly 99.9% identical. Only about 0.1% of our genetic makeup differs from one person to the next. Isn't that kind of crazy? Humans have 46 chromosomes, goldfish have 94, dogs have 78, and those green plants called ferns can have up to 1,020. So do not think that the number of chromosomes an organism has anything to do with its complexity, because that is not true. We have fewer chromosomes than many other organisms, but are considered more complex. Identical twins do not have identical fingerprints. And that is because, did you know you actually get your fingerprints from touching the inside of the womb when you're inside your mother developing? It's kind of interesting, and it causes the membranes on your fingers to change, which causes your fingerprints to be different. So identical twins do not have the same fingerprints. Humans actually share genes with bananas. And if the total DNA in one person were laid out in a straight line, it would stretch to the sun and back over 600 times. That's crazy. That's just a little bit of fun facts to get us started. So, you yourself started as a cell smaller than a period at the end of a sentence. So you yourself, at the very beginning of life, were smaller than the period at the end of a sentence. But now, you are you. You've grown so much. So what happens is when the sperm and the egg come together, you become a zygote. Zygote is a fertilized egg, and then that zygote is going to divide and divide and divide and divide. And division happens many, 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 many times until you are you. So why cells divide? Well, cells divide for cell reproduction, creating clones like in bacteria. So cell reproduction. Growth. We had to grow from a fertilized egg to who we are today. You have to grow from when you're a baby to who you are today. You have to grow. And organisms also need to be able to repair injuries. So if you scrape your leg, you need to be able to repair that. And some organisms do it a bit differently, like some species of lizards can grow back their tail while humans cannot. We can scab over some images. There you might be able to grow back a little portion of the tip of your finger, possibly, but we cannot grow back whole limbs. So some organisms are a bit different in the way that they can repair. Before a cell can divide, many things have to be copied. Everything has to be copied, including the DNA, all of the organelles, the cell membrane, and the enzymes in the cell. All have to be copied before the cell can divide, because we need the new cells to be functional just like the one it came from. Therefore, everything has to be copied. It needs to be identical. Remember, duplicated chromosomes consist of two sister chromatids They contain identical copies of a chromosome's DNA. And when a cell divides, it goes back into being a single chromosome. So here's a single chromosome. Here's our duplicated copied chromosome. When the cell divides, one copy goes into each new cell. Now we're going to come back to an organelle we talked about a while ago called the centriole. So in animal cell division, only animal cell division, centrioles produce spindle fibers. and spindle fibers are going to help guide and move chromosomes through the stages of cell division. So centrioles are a special organelle that create these little fibers. Here are the little fibers here. They're going to grab hold to the chromosomes and help move it where it needs to go as the cell divides. So an overview of the cell cycle, and then we're going to have an image. We're just going to go through the stages. Stage 1 is called interphase. Interphase has many different parts all on its own. Then a cell goes into prophase. After prophase, then it enters metaphase, then anaphase, then telophase, and lastly, cytokinesis. And we're going to have an image for this. And the image is right here. So interphase makes up 90% of the cell cycle. And it is a very important process. And this process is divided into 3 different phases. Now, interphase is a cell doing many things. It could be a cell doing its everyday job. It could be a cell preparing to divide, so it's copying proteins, it's copying DNA, it's making proteins, it's doing its normal everyday thing, or preparing to divide. So we're going to talk about these a little bit more in depth. So the first phase of interphase is called G1. G1 stands for growth phase 1. Growth phase 1 is when the cell is doing its normal everyday thing, like making proteins and energy and growing. So G1 is the cell doing its normal everyday thing. And on our diagram here, B is G1. So I'll label that on here. After G1, the cell will enter S phase. S phase stands for DNA synthesis. So that is when DNA is copied. So during S phase, DNA is copied. Now this needs to go perfectly for the DNA to have identical copies from the beginning to the end. After the DNA is copied, the cell will then enter G2, which is growth phase 2. During this process, the cell is preparing for division. The cell is going to grow more. It's going to copy all the organelles. It's going to copy the proteins, the enzymes, membranes, and everything that it needs prior to dividing. Now, these 3 phases, G1, S, and G2, all make up interphase. So G1, S, and G2 all make up interphase, which is our diagram here. Now, the way that you can recognize a cell in interphase is because during interphase, the cell has a well-defined nucleus. So the nucleus is well-defined, and DNA is loose. So DNA is loose. It's just kind of floating around. Chromosomes are not visible, and the spindle will appear. So let's see. Nucleus is well defined. Nothing goes there. So in interphase, the nucleus is well defined, very clear, loose DNA, there's no chromosomes. That's the big key, is if you see a nucleus and you don't see little X structures, it's interphase. but you're going to start to see the centrosomes here. All right. After interphase, the cell is going to move into mitosis. Mitosis is going to be the division part of the cell cycle. Now, one thing to be clear about, mitosis is nuclear division. The nucleus divides in mitosis, not the cell. So in mitosis, the nucleus divides. The definition of mitosis is nuclear division. The nucleus divides, not the cell. So mitosis, nucleus divides. Okay, now mitosis is divided into 4 smaller phases, and those are prophase, metaphase, anaphase, and telophase. For these, all you need to remember is PMAT. They go in that order: prophase, metaphase, anaphase, telophase. And we're going to talk about each here in just a second. But first, I want to finish labeling this diagram here. So this section here is going to be mitosis. And mitosis has 4 stages. It's kind of cut off with this labeling here, but it has prophase, metaphase, anaphase, and telophase. So we had interphase followed by mitosis. That's where we currently are. Now, the stages of mitosis, we're going to talk about them in detail. So first is prophase. During prophase, DNA condenses, meaning we now have our cell. Our nucleus starts to disappear, so now it's dotted. So we now have our nucleus starting to disappear. We start to see our chromosomes form. We see our centrioles and spindle fibers forming in prophase. So again, prophase, DNA condenses. We now see chromosomes. So chromosomes are now visible. We see our X-shaped structures. Centrioles begin to start moving to opposite poles. You see your spindle fibers. The nucleolus disappears, and the nuclear membrane starts to break down. So the nucleus is starting to disappear. We see our X chromosome structures, our spindle fibers. That's the telltale sign of prophase. Nucleus starting to disappear, and we see our chromosomes. So those are the main 2 parts of prophase. Nucleus starting to disappear, and we see chromosomes. So that is prophase. Then the cell's going to move into metaphase. During metaphase, chromosomes line up single file along the middle of the cell. M for middle. Meta for middle. And this helps to ensure that chromosomes separate properly. So first they have to line up in the middle so that they can separate properly and evenly, so that each new cell, each new nucleus only receives 1 copy of each chromosome. So this will look like, here's my cell. My chromosomes are lining up in the middle. The spindle fibers are attached to them. So they are lined up in the middle. spindle fibers attached to them. Now notice my middle is up and down. It can also be side to side like you see in the PowerPoint image. It doesn't matter as long as it's across the middle. So meta, M, middle. After metaphase, the cell is going to move into anaphase. During anaphase, the spindle fibers are going to constrict. And that's going to make the sister chromatids separate. They're going to become individual chromosomes. The cell is going to start to elongate or get a little longer. And the sister chromatids are going to be pulled to opposite ends of the cell. In anaphase, A4 apart. So A equals apart. So let's draw it out. Our cell starts to elongate. My chromosome is now pulled apart. So now I have two individual chromosomes because I pulled it apart. If anaphase does not happen, this is what's going, if it doesn't happen perfectly, this is going to lead to those non-disjunction mutations, like a trisomy or a monosomy. Okay, so after we pull them apart in anaphase, the cell goes into telophase. In telophase, chromosomes arrive at the opposite ends of the cell. Two nuclei and nucleoli are formed. Chromosomes are going to unwind, and they're no longer visible. The spindle fibers are going to disappear, and cytokinesis begins. So in telophase, let me look at my last one so I'd be sure I did it right. Telophase, T for 2, you start to see two cells. You have two nuclei. Our DNA is not in chromosomes anymore. It's more like squiggles. Now, one thing I want to note, remember when we started this, we said mitosis is the nucleus dividing. The reason that is because this is the last step of mitosis, right here in this image. In this image, I do not have two cells, not yet. They haven't split, but I do have two nuclei. So there are two nuclei in telophase. So the nucleus divided in mitosis. Not the cell. So I have two nuclei. So mitosis is the nucleus dividing. Our last step of the cell cycle is a process called cytokinesis. Cytokinesis is the process of the cytoplasm dividing. The cell actually divides in cytokinesis. So cytokinesis is cell division. And if we wanted to go back up to our original cell cycle diagram, this area here is cytokinesis. And that completes the process. Interphase, mitosis, and then cytokinesis. So the cell grows, copies DNA, grows some more, copies organelles, nucleus divides, cell divides. You start again. So that is the process of the cell cycle. Now, cytokinesis, a bit more on just cytokinesis, it happens differently in plants and animals. In animal cells, there is a constriction around the equator or center of the cell. And this is called the cleavage furrow. And that's going to split the cell in two. It's almost like a rubber band getting tighter and tighter and tighter and tighter until the cell pinches in two. Kind of like you've seen those videos of people putting as many rubber bands as they can around a watermelon and eventually it'll split, it'll burst. Similar thing is happening here. The band around it is just getting tighter and tighter and tighter until it actually pinches in two. But plant cells can't do that. Plant cells have a cell wall. So plant cells, because they have a cell wall, they are not as flexible. And they can't pinch into two. So instead of that, they're going to have vesicles line up in the middle of the cell, and it's going to form what is called the cell plate. And essentially what it's going to do is the cell plate is going to form a brand new cell wall right in the middle of the plant cell. So if this is my plant cell, after I have two nuclei in my one plant cell, there's going to be a form of the cell plate, and that cell plate is going to build a new cell wall. And now I have two cells. So plant cells can't pinch because they have a cell wall. Therefore, they're just going to build a cell wall right down the middle, and then they'll have two cells. Now, this process doesn't just happen randomly. It has to be regulated, because if something goes wrong, it can be very detrimental. So, we're going to talk a little bit about control of the cell cycle. So the cell cycle is regulated by feedback information loops. So in eukaryotes, organisms with a nucleus, the cell cycle is controlled by proteins and has several checkpoints. And one checkpoint is going to be at the end of G1. And if everything is perfect at the end of G1, then the cell will move into cell division, or S phase. After S phase, the cell will be checked again, because S phase is when you copy your DNA. You need to make sure that happened correctly. So after S phase, it's checked again. If it checks out, it'll move to the next step. And then after mitosis, the cell is checked again to make sure it did everything properly. If at any of these checkpoints something is wrong, if the cell notices something is wrong, cell division will be terminated and the cell will be killed. And it does that to prevent mutations, to prevent bad things from happening. Now, sometimes this process goes wrong. Sometimes checkpoints in the cell cycle don't go as planned. Sometimes something went wrong and it's not noticed. And that is what leads to cancer, is when cell division happens uncontrollably. So cancer is a disease in which cells grow and divide at an abnormally high rate. And this is going to happen because there's some type of mutation that triggers this type of growth, or if there's damage to the cell cycle checkpoints. So damage to DNA, which are called mutations, can cause this to happen. It can occur in almost any area of the body, including lungs, breast, skin, stomach, pancreas, colon, heart, brain. It can occur anywhere, and it's kind of scary to think about. Cancer causes tumors. So when cell division occurs at an abnormally high rate, it's going to lead to a growth or a mass of cells. And that mass of cells is called a tumor. And there are two types of tumors. The first type is called a benign tumor. A benign tumor is a tumor that cannot spread. So it cannot spread. And this is typically the type of tumor that can be removed and cured because it's not spreading to other parts of the body. The only time benign tumors can cause trouble is if it gets so big that it interferes with other parts of the body, or if it happens in a very sensitive area like the brain. So if it happens in a certain area of the brain, it could be very hard to remove without damaging something. Now, when most people think of cancer, they think of malignant cancer. Malignant cancer happens when you have a tumor that is capable of spreading. So what would happen is a cell or two from the tumor would branch off, get in the bloodstream, and travel somewhere else in the body. And that can cause a second tumor somewhere else. And the reason that this malignant type of tumor is very difficult to treat is because you don't necessarily know where the cell has spread to. So if a doctor finds a tumor in, let's just say, the intestine, they could take it out, they can test it, and if it turns out to be a malignant type of cancer, they don't necessarily know where it's spread to because, yes, they could scan the body and look for other tumors and try to remove those as well, but if bits of the tumor are scattered throughout and it hasn't started to grow yet, they don't necessarily know where those cells lie. They could be anywhere and everywhere. And that's when it becomes very, very dangerous. All right, so that was a bit about cell cycle and mitosis, what happens when it goes wrong. So we're going to have some practice on Canvas. So go ahead and check